Hi, good evening, good afternoon, good morning, wherever you are. I'd like to welcome you to this special IACH webinar dedicated to the most recent updates concerning multiple myeloma from the ASCO and EHA 2021 congresses. I'm Mohahamd Mohty from the Sorbonne University and Saint Antoine Hospital in Paris, in France, and it is my great pleasure to deliver today this summary of the highlights which was selected by the IACH Scientific Steering Committee. As it is usually the tradition, I'll start with the data in the frontline transplant eligible multiple myeloma patient. And these congresses were, I think, dominated by the updates from the so-called FORTE trial. This trial has been reported already several times in different meetings by Dr. Francesca Gay from Italy. You can see the design of this trial. It is a 3-arms trial, a sort of a non-transplant arm with KRd, carfilzomib, lenalidomide, dexamethasone, and 2 arms with transplantation, but with 2 different inductions, KCD, carfilzomib, cyclophosphamide, dexamethasone, or KRd, carfilzomib, lenalidomide, dexamethasone. Then there is a second randomization in this trial in relation with the maintenance part and testing, obviously, the feasibility of a doublet maintenance with carfilzomib and lenalidomide. You can see here with a longer follow-up the updates from the PFS after the first randomization. Actually, the use of KRd for induction followed by auto is definitely improving progression-free survival in these patients. In addition, the use of doublet maintenance with carfilzomib and lenalidomide is also improving progression-free survival. And when you look to the different risk groups, whether standard risk, high risk, or double hit, we can clearly see the superiority of the KRd auto transplant arm. And we have seen also an analysis of the MRD status at 1 year, especially sustained 1-year MRD negativity in the high-risk patient. And again, the conclusion was that this doublet maintenance combination can lead to a very high rate proportion of 1-year sustained MRD negativity in the KRd auto transplant arm. When you look to the PFS in the different subgroups, and this is from randomization number 2, so looking into the maintenance part actually, and you can appreciate that the doublet maintenance can improve the PFS in all high-risk characteristics, actually according to the authors, except in the amplification 1Q, which remains really a very difficult, hard to treat population. And that brings me actually to a couple of other trials which are now focused on the high-risk population. As you all probably know, the high-risk multiple myeloma population has been traditionally an unmet medical need. Of course, with new drugs, we have seen improvement in this population of high-risk myeloma. However, these improvements did not allow to reach the same outcome at the standard risk multiple myeloma. So it's very good news to see now trials focusing on this subgroup. One of them is this OPTIMUM MUKnine trial from the United Kingdom, reported by Dr. Kaiser and colleagues. And in this trial, actually, they have attempted to have a very strict and clear definition of what we would call ultra high risk patient. And you can see the definition here on the screen. But also from a therapeutic point of view, they have tried actually to put all active drugs together to try to improve the response in this population. 6 cycles of induction with daratumumab, cyclophosphamide, and VRD, bortezomib, lenalidomide, dexamethasone, followed by transplant, followed by 6 cycles of consolidation, another consolidation number 2, but also maintenance. So it's sort of a full, strong treatment package, sort of a total modern therapy, I would say, if you remember total therapy from Dr. Barloghi 20 years ago. And here, what they could see in this high-risk population, the overall response rate is really very high, more than 95% or 90%. And when you look to the MRD negativity results, again, these are really very impressive results, I would say. And these are really important results because we know, at least from data from Spain, that MRD negativity may allow to overcome the high-risk features that you would see at baseline. So this is really good news, and the trial is showing that by putting a strong combination of drugs, DARA CVRD plus V-augmented transplant, you can induce deep remissions in patients with ultra-high-risk multiple myeloma, and actually the safety profile is relatively good because there are no specific safety signals compared to what we usually see when using these agents in this transplant procedure. And that brings me actually to the GMMG-CONCEPT trial. This is a phase 2 trial in Germany, 20 German centers. And this is, again, specifically focused on the high-risk population and using another anti-CD38 antibody, namely Isa-Tuximab. And instead of VRD, like in the previous British trial, they're using KRD, carfilzomib, lenalidomide, dexamethasone, bearing in mind that carfilzomib is a second-generation proteasome inhibitor, probably more potent than bortezomib, because it can rescue the bortezomib-resistant patient. And you can see the schema of the trial putting together all of these highly active drugs, and you can reach up to 100% of an overall response, including a lot of CR and stringent CR, roughly half of the patient. And when you focus on the group of patients with MRD negativity, again, there is a significant proportion of MRD negativity without specific serious safety issues or adverse events. So more and more, I think the data from this concept trial, data from the FORTE trial, data from the UK trials are actually more and more suggesting that combining an anti-CD38 antibody plus a triplet proteasome inhibitor, second-generation proteasome inhibitor, lenalidomide and dexamethasone, should or can become a standard of care in this high-risk population. And of course, this is work in progress, and hopefully we believe that these results will continue to improve with the introduction into earlier lines of therapy of immune therapies like CAR T cells and bispecific antibodies. But I'll come back to this later on in this presentation. Last but not least, in this transplant chapter, I would like to highlight the results of the part 2 from the Cassiope trial, which were reported for the first time during this meeting. I think you're familiar with the Cassiope trial, published already in the Lancet a couple of years ago, and it allowed the approval of daratumumab VTD, bortezomib, thalidomide, dexamethasone, as a standard of care for induction prior to auto-transplant. In this part 2, actually in this trial, we had a randomization between daratumumab maintenance and observation. And here, Professor Moreau could show that actually daratumumab maintenance can significantly improve progression-free survival from second randomization if compared to observation. However, when you dig a little bit deeper in the different subgroups, in the different treatment arms in this protocol, actually the benefit of daratumumab maintenance is mainly seen versus the observation group of patients treated with VTD induction and consolidation alone. So I think this is a very nice study establishing the feasibility of daratumumab maintenance. But of course, for the time being, lenalidomide remains the standard of care, and actually maybe the progress will come from combination trials looking into daratumumab plus lenalidomide or proteasome inhibitors plus lenalidomide. So hopefully in the next few years, we'll see more and more results about the evolution of the maintenance treatment after transplantation. Finally, when it comes to the maintenance, I wanted to highlight the results of the so-called LYRA trial. This is daratumumab plus CyBar-D. Obviously, this is not randomized. As far as I know, this is not approved for induction, but we know that CyBar-D or VCD is a very popular regimen worldwide because of cost issues, because of feasibility. So I thought that we needed to highlight this study because it can be useful for clinical impact. And here in this updated analysis, we know that daratumumab plus CyBar-D induction followed by monthly daratumumab monotherapy maintenance can allow also to achieve deep and durable responses in multiple myeloma. Let me move now to the elderly population, or the so-called transplant non-eligible multiple myeloma population. And actually for me, the recent news are totally, I think, dominated by the updated results of the MAIA trial. So the MAIA trial has been published in the New England Journal of Medicine. This is a trial which demonstrated the combination of daratumumab plus lenalidomide and dexamethasone as a standard of care in the elderly population compared to the doublet older standard of care, lenalidomide and dexamethasone. And we've seen different follow-ups of this trial. And during EHA 2021, we had a late-breaking abstract about the overall survival evaluation. And this is where the results are rather impressive because we have a 60 months overall survival rate improvement in favor of the DARA RevDex arm compared to RevDex. So not only we have a PFS, which is now projected to be close to 60 months, 5 years in an elderly population, but still we have an overall survival advantage. And what's really interesting is that at least 40% of the patients, according to the investigators, were able to receive daratumumab at a time of salvage in the control arm. So in another word, giving the best treatment from the beginning in the elderly population would allow you also to achieve an overall survival advantage. And I think these results, these overall survival results are clearly in favor of using DARA Lindex in the non-transplant eligible multiple myeloma patient. Let me move now to the treatment option in the relapsed refractory multiple myeloma. And I will divide this into two major chapters: the conventional therapies and the immune therapies, including CAR T cells and bispecific antibodies. When it comes to conventional therapies, I think the field is still focused on how to manage the lenalidomide refractory patient. We know that lenalidomide maintenance after transplant is given continuously until progression, so patient will become refractory. I've just mentioned that thanks to the MAIA trial, lenalidomide is also now given continuously, and at some point, the patient will become refractory to lenalidomide. The question is about how to salvage these patients. One option tested in the so-called DARIA trial is the combination of daratumumab, ixazomib, dexamethasone. This is quite attractive because ixazomib is an oral proteasome inhibitor, easy to use, relatively very safe. Daratumumab subcut can be also used, and the results are proving to be quite attractive, showing safety and efficacy. When it comes to the randomized trials, actually, we have now additional analysis from the Apollo trial. So this is a trial testing daratumumab, pomalidomide, dexamethasone versus pomalidomide, dexamethasone. And we know that pomalidomide can probably overcome resistance to lenalidomide. And this has been published. And here we have seen some nice quality of life assessments as part of this Apollo trial, showing that the addition of daratumumab, although it is IV or subcut, actually there is no change. So combining in terms of quality of life for the patient. So combining an oral drug plus an antibody actually is definitely very attractive when it comes to quality of life. And we have also, for the sake of fairness, I need to highlight, we have also the combination of Isa-KRd and pomalidomide plus dexamethasone, which is already approved and widely used. And also we have quality of life data showing that combining an anti-CD38 plus pomalidomide and dexamethasone is allowing for a good quality of life. The other option when it comes to the lenalidomide refractory patient is to use carfilzomib dexamethasone as a backbone and then combine it with an anti-CD38 antibody. This is the CANDOR trial, daratumumab plus carfilzomib dexamethasone versus carfilzomib dexamethasone. It has been reported several times, already published, and now we can see the longer follow-up, more than 2 years, 27 months median follow-up, and you can appreciate that we have a significant advantage in terms of PFS in favor of the triplet combination daratumumab, carfilzomib, dexamethasone. And this is proving beneficial in all treatment subgroups, as you can see on this forest plot. The other option based on carfilzomib and dexamethasone is the IKEMA trial testing Isa-KRd plus carfilzomib dexamethasone compared to carfilzomib dexamethasone. And we had a nice update from this IKEMA trial. And we can see that when it comes to age subgroups, when it comes to high-risk cytogenetics, but also the prior lines of treatments, all of these results are in favor of the combination of Isa-KRd, carfilzomib, and dexamethasone. So if you look today into the landscape of the approved options that we have available to treat the lenalidomide refractory relapsed multiple myeloma patient, we have the daratumumab, pomalidomide, dexamethasone combination, isatuximab, pomalidomide, dexamethasone. We have also daratumumab, carfilzomib, dexamethasone, and isatuximab, carfilzomib, dexamethasone. And all of these 4 trials are leading really to some very good results with an excellent safety profile. But I believe these are not the only options. I think there are also other drugs which can prove to be useful in this lenalidomide refractory population. And here I was pleased to see, for instance, a post-hoc analysis of the Boston trial testing selinexor actually combined with bortezomib dexamethasone versus bortezomib dexamethasone. Obviously, we may argue about the weaknesses of the trial, the control arm, etc. Nevertheless, I think it's a proof of concept that combining selinexor plus a proteasome inhibitor can lead to improved response rate regardless of lenalidomide status. And this is really an important piece of information, especially that we have now the feasibility of selinexor plus carfilzomib and dexamethasone. And this trial has been shown at the last ASCO and EHA meetings. And actually, this combination is quite highly active. It can allow to durable responses, an overall response rate of around 78% with a median PFS of 15 months. So, and these are patients who received a median of 4 lines of prior therapies. So definitely good news for these lenalidomide refractory patients. But the story will not stop here, because we may wish to continue on the IMiD, building on IMiDs, and now we have the new generation of IMiDs now called cell modes, namely iberdomide. And we started hearing now about iberdomide since a couple of years, and here we have an update about the CC 2020 MM001 trial looking into iberdomide plus dexamethasone with daratumumab, bortezomib, or carfilzomib. And these are really very nice results. Of course, we are still at a very early stage, phase 1, so continuous development is needed, but these results are really very promising and paving the way, I think, for the new generation of IMiDs or cell modes. My second part in the relapse refractory setting is about CAR T cells and immune therapies. And here we had some sub-analysis and updates about the KarMMa trial. This is the registration trial, which was reported and published already in the New England Journal of Medicine. This is testing the ID cell construct previously known as BB2121, and this has been approved by FDA, but also by EMA, and is available in different places on a compassionate use in a compassionate use program. And actually, what we can see from the long-term results of this KarMMa trial, that the results continue to demonstrate frequent, deep, and durable responses in these heavily pre-treated population. Remember, they have received a median of 6 lines of prior therapy, and the safety profile is definitely favorable. The other major news from the ASCO and the EHA 2021 meetings is related to the update about CARTITUDE-1 trial, and this is the trial testing the other construct, cilta-cel. And actually, this is now proving to be very impressive, very high number of patients treated, 97, and an overall response rate of almost 98%. Very quick median time to first response, 1 month, and median time to the best response of less than 3 months. And the key message about this CARTITUDE-1 trial and the efficacy of this cilta-cel product is here on this PFS curve, showing a median PFS of 22.8 months, and the median PFS is not yet reached in those patients with stringent CR. So these numbers are very, very high if you compare them to the results of other CAR T-cell construct. Obviously, it's very difficult to compare across trials, but I think this is very exciting to see such a high level of PFS, median PFS, in patients who received a median of 6 lines of prior therapies. And actually, there may be some good explanation from a biological standpoint why the results of cilta-cel are improved, and it has to do maybe with the binding capacity of this CAR construct. So we need to watch out for this new player. But the story of CAR T cells in myeloma will continue, I think, to develop. We have now fully human CAR T cells like this CT103A, and we've seen here relatively short follow-up, a small study, but definitely I think this is probably the future to have fully human CAR T cells, but also maybe dual CAR T cells. Remember, until recently, all the CAR T cell constructs are directed against one tumor antigen, namely BCMA, the B cell maturation antigen. However, we know that one way of improving the results is to have dual targeting. And CD19 is one option, but there are other options actually. But remember, the first case report about the efficacy of CAR T cells in multiple myeloma published in the New England Journal of Medicine was about actually CD19 directed CAR T cells. So here we have a proof of concept that dual CAR T cells can be used with very promising activity. It's not only about autologous CAR T cells, because we have now an ongoing development in terms of allogeneic CAR T cells. And I will not spend a lot of time highlighting the value of having an easily available off-the-shelf allogeneic product. So we have a proof of concept. Number of patients are relatively small, but I think I wanted to put this slide as a teaser, I would say, to show the dynamic of research in this field. Obviously, we still need to overcome many feasibility and safety issues, but we know from the acute lymphoblastic leukemia experience, for instance, that allogeneic CAR T cells can be a good option, and further development is needed in this space. Finally, in this immune therapy chapter, I would like to spend a few minutes on the bispecific monoclonal antibodies, or T-cell engagers. In the last meetings, we've seen an amazing number of studies testing different bispecific antibodies. I think now we have more and more consolidated data about some of these products. And here I'd like to emphasize the results of this bispecific antibody called elranatamab. This is a magnetism trial. I will not go through the mechanism of action, of course, of bispecific antibodies, but what's really attractive here in this magnetism trial, that the patient received a median of 8 lines of therapies, including patients who failed CAR T cells, for instance. So this is quite attractive, and you can see the results. A lot of patients were able to enjoy continuous stringent CR or VGPR. So definitely this is a very promising agent, and the development will continue as a monotherapy or in combination of with other agents, because obviously at some point these bispecific antibodies will need to be tested in the earlier lines of therapy. And this is also the case for Teclistamab, which is an anti-BCMA bispecific antibody. So we've seen here updated results of the MajesTEC-1 trial. You can see the design of the study. At the end of the day, these are heavily pretreated, relapsed refractory multiple myeloma patient. We have here a very good overall response rate. You can see more than 20% of CR, almost 17 plus stringent CR. So definitely these are really very attractive data. The follow-up is still relatively short, but I think the safety profile and the efficacy makes this bispecific antibody really a very good candidate, and hopefully we will be able to use it on a wider scale at some point. However, until now, I mentioned the bispecific antibodies which are targeting BCMA. But at some point, patients may become resistant to BCMA as a target, and you may wish to have other targets. This is why we do have already other candidates. For instance, Cevostamab. This is targeting the FcRH5, and this is a very good target in multiple myeloma. The expression is not affected by the number of lines of prior therapy. So the target is still there. And if you look into the overall response rate, again, it is very attractive. And this is also true for another very exciting bispecific antibody, namely Talquetamab. And we have seen during the last EHA and ASCO 2021 meetings, the update about this phase 1 monumental trial. You can see the design of the trial. First of all, when it comes to safety profile, because of course with bispecific antibodies and T-cell engagers, we may be worried about CRS and neurotoxicity. Actually, here, CRS were generally confined to the setup and first full doses, and actually very few cases of grade 3 or 4 CRS, and the majority of the patients were grade 1 and 2 CRS, and this was manageable with standard supportive care. What's really interesting is the high rate of overall response. So you can see some patients are achieving CR, stringent CR, and a big group of patients, 50%, are achieving VGPR. And these responses were durable and could deepen over time. So hopefully we'll see more and more data about these bispecific antibodies. But now when we look into the huge evolutions in the frontline setting, but also the numerous options we have in the relapse refractory setting, I think patients have more and more options, and this is really very, very good news. So with this, I'd like to thank you all for your attention, for your support to the IACH activities. I hope you have enjoyed this summary from the last ASCO and EHA 2021 meetings. Of course, for the sake of time, the committee could not select all available or all presented data. Some of it was really very exciting. So we had to focus on the most, I would say, impressive results, which can have very rapidly some impact on clinical practice and the way we manage patients on a daily basis. So again, thank you very much. I hope you have enjoyed it, and I hope you will enjoy a wonderful summertime. And please stay tuned for more and more exciting IACH activities. Thank you very much.